All right, good evening, amazing AP Bio 2 kiddos. It's Miss Lynham again, and with today's lecture, we're going to talk about enzymes. And you should know a little bit about them because we have already completed an enzyme catalysis lab, so I just want to make sure to talk about some of the functions of enzymes so that you guys understand what these are. Okay, so the first word that we want to look at is that an enzyme is a catalyst. Okay, if you remember from freshman year, I like to talk about catalysts are things that speed up the rate of a chemical reaction. We talked about your parents being catalysts because they're the ones that make you do your homework, and if you don't do your homework, then they take away, cell phones, and they make you they don't allow you to go out with your friends on Friday night, so it kind of makes you do the reaction quicker, which would be doing your homework. So I like to talk about your parents being your catalyst. They speed up the rate at which you would do a chemical reaction. But in reality, enzymes in the body, these are proteins, okay? So let's not forget what biomolecule group we are under. Okay, these are proteins, and they speed up the rate of a reaction. Okay, how do they do that? It talks about the fact that they lower the activation energy. Okay, this term, activation energy, is the amount of energy needed to get the reaction going. Okay, so the reaction is going to occur normally. It's not like it's a magical thing that's going to happen. So enzymes can only catalyze reactions that would normally occur. So all they're going to do is lower the amount of activation energy that's needed to get that reaction started. Okay, so let's give a few examples of things that are not necessarily biological in the body. Okay, so let's say I had a ball, okay, and here's the ball, and it's sitting in a little divot on a hill. Okay, so here's my hill. And I want this ball to be able to go up and over and go down the hill. So I want it to be able to roll down. As you can tell from this diagram, in order for me to get that ball up and over, it's kind of equivalent to like the roller coaster effect. In order to get it up to that point, it's going to take an investment of energy. I'm going to have to use some energy to get that up to that transition state, at which point then my reaction will occur. The same thing is true if I talk about enzymes. Okay, so look over here in the diagram. In this picture, I have A, okay, which is considered to be a reactant, that is bonded to B. I've got C that is bonded to D. Okay, look at what my product should be over here. Okay, in my product section, I want to have A bonded to C and B bonded to D. So I'm going to need to switch up these bonds and have a whole different product down here at the end. Okay, so in order for me to do that, I have to reach what is called the transition state. Okay, to reach the transition state. So in order to do that, there is a certain amount, here's the circle right here, of activation energy. It's E sub A. Okay, the E is energy, A is activation. So it's going to take a certain amount of activation energy to get that reaction to happen and get to my products over here. Okay, a perfect example besides the ball would be if you guys think about fireworks. You guys probably all go to fireworks displays on the 4th of July. And so with fireworks, you've got fireworks that go off and have to react with oxygen. Okay, you might not know this, but the products from that, you get carbon dioxide and you get water. Okay, if you think about the reaction. So I've got fireworks over here on this side, plus oxygen yields carbon dioxide and water as my products. But you've got to have a spark that makes that firework go off. You've got to light it with a lighter. You've got to have that igniter part. Well, that's an example of you have to have this input of energy to reach the transition state in order to then make the reaction happen. Now over here on this side, okay, my products, I'm going to have free energy available. Okay, so you can see the free energy, this is Gibbs free energy, is less than 0 whenever I have my products. Well, what an enzyme does, okay, here's what an enzyme does, it lowers this arrow. It lowers how much energy is needed to make the transition happen to then make my reaction happen. So an enzyme lowers this activation energy, so I don't need as much of a spark to get the reaction going. So it's really, the enzyme is working on the rate. That's what an enzyme is working on, the rate of a reaction. Okay, if you read in your book, a perfect example was talking about sucrose. Okay, and I know I haven't talked about the carbohydrates yet, but sucrose is a disaccharide. Okay, it's a disaccharide. It's 2 sugars bonded together. In fact, it is glucose, glu plus fructose. That's what sucrose is. So in order for your body to metabolize this, like if you ate a spoonful of sugar, your body would have to break the covalent bond between the glucose and fructose molecules. Well, I don't know if you read in your book, but it's pretty amazing that if you ate only one of these disaccharides and you did not have any enzymes available, it would take you 15 days to break up that one disaccharide into glucose and fructose. 15 days for one. Okay, with the enzyme, okay, and the enzyme's name sounds like it's breaking down. Here's the enzyme, is called sucrase. With the enzyme sucrase, it only takes 1 second for your body to break down one sucrose molecule. So there's a significant difference in the rate of the reaction due to the presence of the enzymes. Okay, and I want to show you really quickly that ASE, anytime a word ends in ASE, that normally refers to an enzyme, and it's going to talk about in the beginning of the word, what is it working on. Okay, so there's another one called lactase. Can you guys guess what that works on? Okay, hopefully you said lactose, because you're correct. And there's another one called maltase. Can you guess what that works on? Hopefully you said maltose. So we have these enzymes in the body that end in ASE that work on certain reactions. Okay? All right, let's go to the next slide. See if we can get to the next one. Okay, here's a perfect diagram where you can see the overall impact of the enzyme. Okay, so you can look at the graph where you see the course of the reaction without the enzyme. Look at the black line. Okay, so here's my activation energy, my black line right here. So I'm going to need this amount of energy to get that reaction started. Okay, look at my products. I'm going to end up being at this location right here, so I'll have my products at the end. So this reaction is still going to happen on its own, even without an enzyme, but this amount of activation energy is a lot higher. Okay, if I've got an enzyme present, look at it doesn't take very much to get that ball rolling over the hill. So my activation energy is lower, but look what I end up with over here. I still have the same amount of free energy at the end of the reaction. So the free energy is unaffected by an enzyme. It's not doing anything to the energy at the end. What it's doing is lowering the beginning, the activation energy, that transition state, how much energy it takes to get over that hump, therefore speeding up the rate of the reaction. Okay? All right. Hopefully you got that. Okay, let's talk a little bit more about enzymes and their abilities and what they do. So there's a few bullet points on here that I want to talk about. Okay, the first word is recycled. The great thing about an enzyme is they are never used up. Not only are they never used up, they're not changed by the reaction. The same enzyme can be used over and over, okay, over and over again. You can keep using the same enzyme. Okay, there's a few little stipulations though. Enzymes are temperature and pH sensitive. Okay, I'll give you an example that you probably remember from freshman year when we talked about digestion. There's an enzyme called pepsin, which is found in the stomach, and it only works in a range with a pH of about 2. Okay, it's actually about a 1 to 2, and so that's a very, very acidic environment, a 1 to 2. If you take pepsin and you alter the pH environment, okay, you bring your stomach content up to, let's say, a 5, which is a weaker acid, that's going to impact the rate at which pepsin, the enzyme, can do its job. And pepsin's job is to break down proteins. Okay, temperatures are, this is another example. Enzymes work best within a certain range. Okay, a lot of the enzymes in our body work best between like 95 and 99 degrees. If you break homeostasis and you have a fever or you have something wrong with the body, maybe you're going hypothermia the other direction, that can impact the overall rate at which enzymes can function. Okay? The third little bullet here is enzymes are substrate specific. Okay, substrate specific, meaning that an enzyme usually only has one reactant. Okay, we call that the substrate, one reactant slash substrate, or maybe it's got two, can't spell, maybe it's got two substrates, but they're very closely related to each other. Okay, so in the case that I gave you a minute ago, sucrase is specific to work on sucrose. Okay, sucrase works on sucrose. Maltase works on maltose. Lactase works on lactose. So substrate specific means that each enzyme is designed to fit to one reactant. It's going to bind to one reactant or one substrate. Okay, another point about enzymes is sometimes they do require a cofactor or a coenzyme in order for them to be functional. So sometimes they have to take like an activator molecule to turn them on, and that is what would make them be able to catalyze reactions. Okay, so if you look at the diagram down below, let's talk about this for a minute. We're going to label this. This is our enzyme. Okay, that is enzyme, is the big purple blob. Okay, this thing that is coming in, this is going to be our substrate. Okay, so if I tell you, for example, that this is sucrose, it's a disaccharide, and it's linked by a covalent bond right here. So it was linked, remember, through dehydration synthesis. Okay, so this substrate, sucrose, comes in and it fits to the enzyme in this place right here that I'm outlining. That location that I'm outlining on the enzyme, so you can see it right here, right there, is called the active site. Okay, active site. It's the place on the enzyme where that substrate can bind to. Okay, now in this diagram you see, we can't forget, we're going to need some water. Because in this reaction, we are hydrolyzing. We're doing a little hydrolysis. Because look at my products over here at the end. They are split. They're broken up. So this enzyme is hydrolyzing. It's speeding up the rate at which hydrolysis is going to break up sucrose. These two products over here, one of these is glucose and one is fructose. So I'm going to need a little bit of water because it's hydrolysis. In order to break, I'll change the color so you can see, I'll use a black here, so I can break that covalent bond. Okay, so the active site, once again, is this place on the enzyme at which the substrate binds and the action is happening. Okay, so in this diagram, this one, there's the bond is broken, now I have my products on this end. Okay, so my sucrose substrate slash reactant, that's what comes in. What I have at the end are my products. In this reaction, I hydrolyzed or broke the bond, so I need a little bit of water. Notice as this arrow comes around, my enzyme can be used over and over and over again. Okay, they're recycled, never used up. Okay, next thing. Okay, I wanted to mention one thing here before I forget, guys, on this place of that active site. I want to make sure and mention to you that the specificity, okay, go back up to this word, substrate specific. Okay, up here, if my pen will work, here we go, substrate specific. That specificity results from the 3D shape, okay, that specificity results from the 3D shape and the chemical properties of that active site. So this active site has a specific shape that is three-dimensional that helps determine what substrate it's going to fit to. Okay, the other thing I wanted to mention is when this, in this case, sucrose that I was talking about, when it comes in and it binds to that active site, okay, this is known as the ES complex. Okay, look at the ES. It's enzyme substrate complex. So whenever you see the substrate bonded, in this case to the enzyme, this is known as the ES complex. Okay, I wanted to mention here that the bonds that hold that together, that hold the substrate into the enzyme, it could be different types of bonds. Sometimes it's hydrogen bonds that hold that together, which would be, remember, a weak attraction between a positively charged hydrogen and something else that's electronegative. It could be temporary, okay, temporary because that's only going to be for a brief time, covalent bonding that could hold that in place, or it could also be an electrical attraction. So that substrate is going to bond into the active site of the enzyme either through hydrogen bonding, temporary covalent bonds, or electrical attraction until the bond, in this case hydrolysis, could be broken. So that's my ES complex in order for that to happen. It's having this enzyme substrate complex. Okay, does that make sense? All right, let's go to the next one. And I love the description of this one because for years and years we have taught this as what is known as the lock and key method. You might have actually heard of that before. Okay, let me go back one because I just got off that. You might have heard of the fit of an enzyme being known as the lock and key. I love the new description in all the textbooks because we don't teach it as lock and key anymore. It's literally like a catcher's mitt, okay, a catcher's mitt to a baseball or a softball. Okay, so think about a catcher. When a pitcher is throwing the ball to the catcher, the mitt is shaped a certain way, but when that ball slams into the mitt, the catcher like squeezes the mitt, and then at that point the mitt fits around the ball perfectly. That's this idea of an induced fit of an enzyme. It's a perfect example. So as the enzyme envelops or surrounds the substrate, there is a slight change that takes place in those bond angles in the active site. Okay, so whether it's hydrogen bonding, whether it's covalent bonding or electrical attraction, there's a slight change. There's like a wiggle, like I like to think of it, like a wiggling in the mitt. And at that point, the atoms can be oriented between the substrate and the enzyme. The atoms face each other, and then the chemical groups on the side chains of the amino acids of the enzyme can interact with the substrate. So you've got that chemical interaction to happen between the amino acids that are in the enzyme to the atoms that are in that substrate. That allows the chemical reaction to occur more readily. So that's the job of an enzyme, guys. The whole purpose of it is to bring together the specific atoms so that the bonds can form between them. Okay, so it helps to do that. So we call this an induced fit. And that was one of the questions, by the way, on your pre-lab quiz. That was the 5th question that a lot of you guys missed. So I wanted to mention that. So it's not a perfect come up immediately as you see in this picture. These are not going to come in perfectly right away. There's going to be a wiggle of this enzyme around the substrate. Okay, and in this case, look what we have at the end, by the way. This is not hydrolysis. This diagram is showing dehydration synthesis because I have 2 monomers coming in, and look at my product over here. I have, in this case, let's say it's a disaccharide as a result. So I had 2 monomers come in, maybe this is glucose this case, and this is fructose come in, and they are going to bond, okay, after the induced fit. They bond together, there's my covalent bond, and then I have my product, which is my disaccharide at the end. So this one is not showing you hydrolysis, it's showing you dehydration synthesis, because enzymes can help speed up both hydrolytic actions and synthesizing, like reactions that are putting things together. Okay, so that was the induced fit. I just want to make sure you knew that it was like a wiggle. It's like a catcher's mitt around a ball. Okay, next thing we're going to talk about, actually before I get to that, I want to talk about one of the graphs that is in the book. Okay, so there was a graph and it talked about rates. And this is going to help you with your lab that you're turning in your enzyme catalysis lab. There's a perfect graph that talks about the relationship between the concentration, okay, the concentration of the substrate, C-R-A-T-E, and the reaction, that's reaction rate. Okay, so let's just say that this is my maximum rate right here. I'm just going to put a line across. Okay, that's that line. Okay, now I'm going to make a color. I'm going to use a blue. I'm going to show you that if there is no enzyme, okay, look how this goes up, the rate of the reaction is proportional to the concentration of the substrate. So, for example, if I have more substrate, okay, so let's just talk about maybe in your lab the H2O2, that's the hydrogen peroxide. If I have more of the substrate, if I have no enzyme, I'm going to put by here, no enzyme present. Okay, look at what happens to the rate, the speed at which the reaction occurs. So if I have a lot of H2O2 and no enzyme, it's going to continue to break down over a long period of time. Okay, but if I introduce an enzyme, okay, watch what happens if I use the enzyme. In the beginning, my reaction rate is going to speed up quickly, but look what happens to my graph as I increase the amount of substrate with an enzyme. Okay, let's talk about that. If I only have, and I'm just making this number up, if I only have 20 enzymes available, okay, 20 enzymes, and here they are available, ready to work. If I increase the substrate, okay, the amount of H2O2, in the beginning, all 20, okay, are going to start filling up their active sites, and as they start working, the rate is going to speed up, like this. But as I increase how much substrate I have, at this point up on the top of the graph, all 20 enzymes are going to be filled up. They're all doing a job. What we say about it is the enzymes are saturated. All of my enzymes that I have available are completely, the active sites are filled up with a substrate. So look at my line. My line levels off because the rate is not going to continue to increase indefinitely with an enzyme because my enzymes are all occupied due to my increased concentration of substrate. So it's kind of a weird looking graph, in this situation because you have to look at how many enzymes are available and how many of them are doing a job. Okay, so that's one way of looking at it. So the no enzyme is going to continue to increase because as we have more substrate, we're continuing to break down, but it's taking a longer period of time. But if we have those enzymes, the rate increases drastically in the beginning until the enzymes are saturated, at which point they level off. And so my max rate is reached at this point rather quickly, and then they reach a rate of saturation. Okay, so I just wanted to make sure I mentioned that. Okay, the next thing we're going to talk about, and we only have like 3 slides left, so we're almost done, hang in there. We're going to talk about inhibition of enzyme function, okay, how enzymes can be inhibited. Just think of the word inhibit. Okay, if they're inhibited, they're going to be impacted and not be able to do their job. So we have 2 types of inhibitors. Okay, look at the words. We have competitive inhibitor and we have non-competitive inhibitor. These are things, molecules, that can make the enzyme stop doing its job, but if it's competitive, it's mimicking the substrate and it's going to come into the active site. So look at my picture over here. Okay, here's my normal substrate. Okay, this blue thing that you see, that's what should be coming into the active site right here to be able to do the job that we need to happen in this case. If it's a competitive inhibitor, like in picture B, this is a molecule right here that comes in and binds to the active site and blocks the substrate from being able to attach and form bonds with the active site of the enzyme. So that's a competitive inhibitor. A non-competitive inhibitor, okay, like in this picture C, is still a weird-looking molecule. It binds to a place not on the active site, so somewhere else on the enzyme. In this case, it's bonding over here to the left side. And when it sticks in, what it does is it changes the shape of the active site. Because if I stick that in here, it's forcing the shape to change as it's breaking up some bonds on that active site. Now my substrate will not fit. So it's not competing with the active site spot. It's pushing on a different part of the enzyme, which then changes the shape of that active site, and now my enzyme can no longer, my substrate can no longer bond with my enzyme. So that's non-competitive. Okay, make sense? All right. Okay, I did want to mention on this slide before I move on, I wanted to mention that both of these inhibitors, okay, that you see here, the competitive and the non-competitive inhibitor, both of them are reversible, okay, reversible. Both of these are reversible. Oh, this pen is driving me crazy. Both are reversible, and in both cases, that inhibitor, okay, this inhibitor molecule right here, is binding non-covalently. Non-covalently, right there, and so that is able to be reversed. So if, let's say, the bonds change in this place, because it's not covalently, it might just be a sticking or a attraction. If this pops out, now my substrate can fit back into the active site. Or in this bottom picture, if this non-competitive inhibitor pops out, then this can loosen up and go back to the normal shape of the active site. Okay, but there are some irreversible, okay, if you read in your book, there are some irreversible inhibitors. S-I-B-O-E. There are some irreversible inhibitors, but what those do, it's interesting, is they will actually covalently bond, okay, covalently bond to an amino acid side chain, okay, so to like an R group that is in the amino acids located right here in this active site. So if they covalently bond to a side chain, an R group, it could be an irreversible type of inhibitor. Okay, so I just want to mention that. Okay, almost done, guys. 2 slides left. So here we go, rock and roll. Okay, the next slide is talking about allosteric regulation. Okay, and we actually just introduced that. I just didn't use the word at that point. So if you look at the word allosteric in Latin, okay, allo, right here, this word allo means different. Stereos, okay, it's actually S-T-E-R-E-O-S. It sounds like stereos. Stereos is shape. So an allosteric regulation is a different shape regulation. It is that example I just showed you of that non-competitive inhibitor. If a molecule binds to an enzyme at another site, meaning not the active site, this is an example of a way to regulate enzyme functions. Your body does this all the time in certain instances. We have molecules that can bond to not the active site, but to a different location, and can regulate enzyme activity. Okay, it can result in the activation of a formerly inactive enzyme or the inactivation of an enzyme. So it's a way for us to turn enzymes on and off, okay, all the time in our bodies. The activator or the inhibitor can attach to the allosteric or the regulatory site. So in this picture, if you look over here, where this activator right here was attached or is coming in this location, we would call this place, this is not the active site, we would call that the allosteric site or the regulatory site. It's where that regulatory molecule would bind to then change the shape of the enzyme, and it could either turn an enzyme on or turn an enzyme off. Okay, so these little activators or inhibitors, in this picture it's showing you the activator right here, these could also be inhibitors that bind. They will attach to that allosteric or regulatory site non-covalently. Okay, so it's gonna bind there for a moment, and it can change the shape of the active site. So that's one example. We will talk about this definitely a lot when we get to cell respiration and photosynthesis, because this can happen by a process known as phosphorylation, and we will talk about phosphorylation and glycolysis. And so it doesn't have to just be this weird activator molecule or an inhibitor. It could actually be the sticking on of a phosphate group. Okay, remember the inorganic phosphate group? Can bond, which could also change the shape of an active site of an enzyme. One very important group that we will hit in this year is we will talk about these things called protein kinases. Okay, protein kinases, and these are a very important class of enzymes that regulate responses to the environment by organisms, and they are in this example of allosteric regulation. They can be turned on and off by other molecules like activators binding to them or inhibitors binding to them. So it's a lot like, I just want to make sure you understand, it's kind of the same thing as this slide. Allosteric regulation is a way of regulating enzyme function by using these non-competitive inhibitors. Okay, so a non-competitive inhibitor can attach to another part of an enzyme, changing it. It's an example of allosteric regulation. I just wanted to make sure I hit on that. Okay, and the final slide, last but not least, is talking about feedback inhibition. I'm sure you remember from either Bio 1, and some of you guys are in anatomy, and it's similar to feedback inhibition in the body. Okay, feedback inhibition. We talk about it with the human body, like let's say your blood sugar level rises, then you would secrete a hormone called insulin. Okay, so you would secrete insulin, which would cause the glucose to decrease in the body, and as soon as the glucose levels come down and the hypothalamus senses that decrease, insulin production is going to decrease at the pancreas. So that's just one example you heard of freshman year. So if you look at how it fits into this slide, feedback inhibition is another way to regulate enzyme functioning by looking at how much end product is being synthesized. So if we have an accumulation of the product, okay, let's say we're talking about we have an accumulation of glucose and fructose by breaking down sucrose. Once that product is produced and it's accumulated, the enzyme can be inactivated or turned off in a certain metabolic pathway. So if you look at this example over here, okay, and I did want to make sure that I mentioned really quickly, a metabolic pathway, we're going to talk about lots of these in class, but they usually involve a start. Okay, let's see if I can get my pen to work this time for the last slide. They usually involve a starting material, a metabolic pathway. Okay, they have various, after that, various intermediate pathways. Okay, so like for example, when we talk about cell respiration, we're going to talk about glycolysis, Krebs cycle, and electron transport chain. That's a metabolic pathway that has something that we start with, various intermediate products along the way, and then after the intermediate products, we eventually have an end result, okay, or an end product. So there are numerous reactions that happen in the body that would take place in this metabolic pathway, each that forms an intermediate. So look at the diagram that you see here. Okay, here's my threonine, okay, right here. There's my enzyme. See my enzyme, and it's showing you the active site available. So if threonine binds here, we're going to have several intermediate pathways that are going to take place. You can see intermediate A, then we've got the presence of enzyme 2. Then we get an intermediate B, and then we have enzyme 3. Intermediate C, then we get enzyme 4, intermediate D, and these are all different forms, okay, different changes. We get enzyme 5 that helps out to form the final product. In this case, it's called isoleucine. Okay, so the first step is known as the commitment step. It's like getting the ball rolling. Over here, the attachment of threonine in the active site, that's the getting the ball rolling. The reactions happen in sequence, leading to my final end product. It's wasteful for the cell to make something that it doesn't need. So if we have enough of this end product, we have tons of this isoleucine, there's no need to keep going down the same pathway. So what happens is this can actually attach. So isoleucine, which is the product, goes up and binds allosterically to the allosteric site, which, look what it does here, changes the shape of the active site, so it then turns off the enzyme. So feedback inhibition is we have enough of this end product, we're going to use that product to bind to the allosteric site and tell the cell, okay, to use it up, and we don't need to convert threonine into this isoleucine anymore. So it's a way to regulate enzyme functioning so that we can get what we need in the body, and we do this all the time with different enzymes. Okay? All right, that concludes enzymes.